Let's start to continue our lectures about respiratory chain and oxidative phosphorylation. In this lecture, we'll be explain how the chemoosmotic theory that we explained in the last lecture, how this theory will be result in coupling oxidations along the electron transport chain linked to phosphorylation, how phosphorylation is occur and how ATP is produced. And also explain what is the function of complex 5 in electron transport chain. Start to taking an overall what is happening in respiratory chain. The respiratory chain begins with reducing equivalent called NADH and FADH2. We wanting to oxidize the final acceptor of electrons is oxygen to forming water. The respiratory chain is present in inner mitochondrial membrane. They are arranged into 4 complexes. The complexes one is called NAD dehydrogenase. The NAD dehydrogenase converting NADH into NAD. And this passing electrons and the proton into QH2. QH2 is a reduced form. The complex 2 it is called succinate dehydrogenase. Succinate dehydrogenase it is a co-prosthetic group FADH2 and iron sulfur protein. We'll be converting hydrogen into Q to forming QH2. Passing from complex 1 to QH2, this resulting in energy. This energy it is used to pumping proton into inner mitochondrial membrane. Proton is carrying positive charge. While passing from complex 2 to the QQ, this resulting in the no free energy enough to pumping proton. And then QH2 will be forming electron, passing electrons to cytochrome C. This is the reduced in the form of carrying hydrogen. This is reduced in the form of carrying electrons. Passing electrons from the QQ to the complex cytochrome C, and this is called through the complex 3. Complex 3, it is called, this is result in reduced cytochrome C, so it is called cytochrome C oxidoreductase. And then the electrons from cytochrome C is passing through oxygen forming water. This is oxidized, complex 4. Oxidized cytochrome C, we call the complex 4 cytochrome oxidase. Passing from the complex 3 to complex 4, this is resulting in increasing energy. This free energy is used to pumping proton to inner mitochondrial inner mitochondrial space. Passing from the complex 4 to oxygen, forming water, also resulting in increasing energy. And this is energy resulting also in pumping proton inner mitochondrial space. As you see here, the proton it is a positive charge. This is positive charge creating a positivity in inner mitochondrial membrane. Regard to the mitochondrial matrix. The high concentration of proton pumping through this passing oxidation reduction in the respiratory chain, this results in increasing amount of proton inner mitochondrial space. When increasing the amount of proton inner mitochondrial space, this has become more positive charge. This is produce electrochemical gradient. Electron positive gradient here. This is proton 1 to 2 passing. Unfortunately, the inner mitochondrial space is impermeable to the proton. No, you can't passing through here. The only way to passing this proton to the mitochondrial matrix to passing through the complex 4, the complex 5. Complex 5 it is called ATP synthesis. Passing this proton here to the complex 5, this is the only way to re-enter again to mitochondrial matrix down high concentration gradient, be resulting in activation this enzyme forming EDP into converting EDP into ATP. Starting from NADH, this resulting in 3 chances of pumping proton here and re-enter and activating ATP synthase and forming 3 molecules of ATP. When I start with the FADH2, it was not starting here. I was starting with FADH2. FADH2, no, unfortunately, no here. In these steps, no passing hydrogen. So only I having 2 chances of pumping hydrogen into inner mitochondrial space, and which is re-enter again through the complex 5 and forming only 2 ATP. This is explain the secret of forming 3 ATP when starting with NADH and forming just only 2 ATP when starting with FADH2. Passing NADH through passing of hydrogen down concentration gradient through the complex 5 and the phosphorus. The chemoosmotic models. What is the chemoosmotic theory? The passing of protons in complex 1 and electrons in the complex 3 and 4 resulting in it is a higher steps in the stairs. It is resulting in releasing of free energy, and this free energy is stored passing from complex 1, complex 3 and 4 resulting in passing of energy. This is energy. It is utilized to pumping electrons in inner membrane space. This theory postulated that this energy obtained from oxidations passing here from hydrogen or from electrons will be resulting in pumping electrons in inner mitochondrial matrix. As you see, the proton is carry positive charge. This generating electrochemical gradient. Why? Because the proton in inner mitochondrial space can't passing again to mitochondrial matrix because the inner mitochondrial, the key point is inner mitochondrial membrane, it is impermeable to proton. This is the proton pumped can be returned again. This generate electrochemical gradient across inner mitochondrial membrane. The outside it is positive charge because of proton relative to the negative charge presenting inside. This proton want to return again. The only pathway to return again to mitochondrial matrix it is occurs through the complex 5. This is complex 5 allowing entry of proton down this concentration gradient that may be resulting in generating energy, and this is energy would be utilized in phosphorylation of ATP into ATP. So this couple this oxidation to the part of phosphorylation in the complex 4. Complex 4 it is called ATP synthesis. Complex form it is forming of the two parts F0 and F1. F0 it contains two domains. F0 it is the span inner mitochondrial space. Space is span the mitochondrial membrane, and the other part which is called F1 it is the protruded into the mitochondrial matrix. The F0 and the F1 containing multiple proteins alpha, beta, gamma and so on. The hydrogen, the proton is passing through the only pathway to return again to mitochondrial matrix to passing through this complex. Through the F0, when it is passing the F0, they are stimulating S rotatory pathway. This is the unit will be rotated. When it is rotated, it will be activated the beta unit of the B1, which is ATP synthase, converting ATP to ATP. The chemoosmotic hypothesis proposed that after protons have been pumped into outside the inner mitochondrial membrane, which is called outside, they called intermembrane space, they want to re-enter the matrix, but the inner mitochondrial membrane it is impermeable to it. The only pathway to the proton enter the mitochondrial matrix it is occurs through the F0 domain. Passing the proton through the F0 domains resulting in rotation down concentration gradient result in rotation of F0. At the same time, they are dissipating the pH electrochemical gradient. What this means by dissipating, this is the remove the concentration gradient. The inner membrane, it is high, containing high proton, so it is creating positively outside to negative. This dissipating, this is the difference in electrical. F0 rotation causing conformational changes in the beta subunit of F1 domain. And this allow ADP to binding into phosphate and forming ATP. This is the formation of phosphorylation. So we couple for oxidation with the bumping of proton to activation of ATP synthase in the F1 domain in the complex 5 to form ATP by phosphorylation. The stepwise production of energy it is occur ATP as the physiological behavior. Why? It is the NADH, the hydrogen is passing directly to oxygen. This will not result in gradual release of proton and the gradual entry through the F complex to the complex 5 with the formation of ATP. This is the normal. So they are gradual release of energy, gradual formation of ATP. It is passing down its concentration gradient from inner membrane space to the mitochondrial matrix through the F0, this resulting in its rotation with activation of the beta unit, which is presenting in F1, causing ATP synthase with the formation of ADP forming ATP. This okay? Manner. The chemoosmotic models, they are supported by many findings. What is the finding? That if we adding proton to external media of the proton, this is what happened by oxidation. Oxidation also produce proton to the external media. When we adding more proton, we stimulating more ATP production, because this proton again will be enter through the complex 5, which is the F0, F1, and stimulating ATP formation. Adding more proton, more ATP will be formed, and the proton result from oxidations through the respiratory chain. At the same time, this is oxidative phosphorylation. It is obstacles. It does not occur if the mitochondrial membrane, inner mitochondrial membrane is soluble to hydrogen. Suppose that we producing sieves of the inner mitochondrial membrane, so the proton will be come to outside, will be enter again, come, will be enter again. So there will be no concentration gradient, no activation of ATP synthase, and no phosphorylation will be occur. So this is two theory confirm the presence of the chemoosmotic models. What is the PO ratio? يعني this is means that how the number of ATP molecules this occur per 1 oxygen molecules is consumed forming water. Per every oxygen forming half of oxygen molecules forming water, this resulting in 3 ATP formation by, roughly speaking, by NADH, while it is concerned to 2 ATP formation by FADH2. But uncouplers producing 0 ATP. What is the uncouplers and how it is produced 0 ATP? Starting from NADH, as we taking before, will be resulting in 3 ATP formation, and this is hydrogen will be formed to oxygen forming water, while the hydrogen or the FADH2 forming water will be resulting in formation of 2 ATP, while the uncouplers 0. What is the uncouplers? Another one. All these correct for CoQ, except which one of these are incorrect about CoQ? Yes, it is accept reducing equivalent from cytochrome A and A3. It is wrong. It is reduced by the complex 1 and 2, yes. Complex 1 and 2, or both of them, converting CoQ into CoQH2, yes. Transfer of electrons to complex 3, yes. The complex 3, complex is a link between complex 1 and 2 to complex 3, and the complex 3 passing in the form of electrons, yes. It is a lipid soluble, yes. It is the mobile, and all part the other is the fixed. It is a carrier proton electrons, yes. It is the QH2. QH2, it is 2 electrons and 2 protons. It is accept? No, it is not accept. It is transfer to the cytochrome AS3, not accept. The accept from cytochrome B and C, which is present in the complex 3. And then transfer, not accept. It is accept, it is the transfer. The correct is transfer it to the complex 4, which is cytochrome A and A3. Another question. Terminal cytochrome of respiratory chain which donate electrons to oxygen is, which one of them is terminal cytochrome, the last cytochrome in respiratory chain to producing electrons to oxygen, which is the final electron acceptor. Yes, of course, it is cytochrome A3. Which one of the following accepting only 1 electrons? Oxidation reduction reaction is occur by accepting electrons or by accepting hydrogen. Hydrogen, it is the 2 electrons and 2 protons. Which one of them is occur by accepting electrons? Yes, it is cytochrome. As we said, cytochrome and the iron. Iron is presented in the cytochrome in the porphyrin, or iron-free iron as iron sulfur. It is accepting, oxidizing, and reducing electrons. While the coenzyme coenzyme Q, FMN, and FAD, and even NAD, all of this, it is not in the form of electrons. Thank you.